Assalamu alaikum. Bismillahirrahmanirrahim. Hello everyone. In this tutorial, we will be talking about the codon optimization. The codon optimization for exotic gene transfer. So, what is basically, first of all, exotic gene? Actually, the exotic means non-native or foreign. It is, in scientific term, it is also called heterologous gene. So it is topic is like codon optimization for heterologous gene transfer. And in this lecture, we will see how to design DNA sequences for optimal soluble protein expression. So basically, what is optimal soluble protein expression? Well, first of all, you should know that what is the protein expression. First of all, I am giving you the basic fundamental knowledge behind the codons and codon optimization, and then at the end we will talk about the technique that how we can optimize the codon. So let's back on the protein expression. Protein expression or recombinant protein expressions refer to the manufacturing of the protein derived from the recombinant DNA or for foreign DNA, or protein expressions can be more specifically explained as or refer as the way in which proteins are synthesized, modified, and regulated in living organisms. Protein expression overview, that we are talking about the heterologous protein expression. And this is the overview I want to give about the protein expression, how we can overexpress our protein. Before going into the overview, I would like to tell that expression of the functional protein in a heterologous host is a cornerstone of the modern biotechnology, because functional proteins are required for the determination of the three-dimensional structure of the protein. It is required as a chemical probe or tool. It can be used as an antibody, hormones, enzymes, or new type of a drugs, among others. So it has a various uses. That's why the modern biotechnology is uh, so much so depends upon the functional expression of the protein. This is called significance or importance. You can see the significance importance of the protein expressions, functional protein expression. So usually in lab, we usually get the functional protein expression by gene cloning. First of all, we should know our gene and ultimately the amino acid sequence. So we select the gene of interest that every enzyme or protein is coded by the DNA. So we should know that gene, for example, the insulin gene. So then we use the expression system. What expression system we are going to use for, because different microbial hosts, they have they use different expression vectors. These expression vectors or expression systems are also called plasmids, because the plasmid has similar promoters, ribosomal binding sites, and terminations like the host. So host can recognize those cloning vectors. Then we construct a clone that gene into the plasmid, and then we transform that plasmid into our host, for example, like E. coli or some other bacteria or microbial host. Then if there is expression, then we can get the protein, and we can purify the protein, because cell has their own protein, thousands of different proteins. So among those 100,000 different proteins, we have to purify our protein of interest or heterologous protein. So there are different methods which we can identify our protein. Once we get the pure uh, protein, then we can characterize, study, and do anything depend upon our study, the purpose of our study, we can do with that. The pure protein and characterization depend on. And we can also, after purification, the protein product can also be analyzed by the STS phase analysis. Actually, it gives the unique band representing to the molecular weight of our proteins. Gene actually expression is influenced by the many different factors, or there are factors in soluble protein expression optimization. Gene cloning and getting protein expression tend to be very simple, or it looks very simple that it is simplified. I will clone the gene and get the protein expression. However, in practice, there are many factors which can influence this process and can make the expression of protein quite challenging. So there are different uh, factors which can need to be addressed for soluble protein expression optimization, and there are different factors which such as rare codons or codon optimization, the stains, the microbial stain used for the protein expression, different factors, protein synthesis rate, growth medium, fusion tags, and co-expression proteins. Co-expression of colonization or rare codons, which we will discuss in detail in this lecture. So I will just briefly discuss about the strains, vectors, protein synthesis, growth medium, fusion tags, and co-expression, just for the basic understanding. So how stains can matter, because there are different microbial stains are used for heterologous gene expression, but the most commonly used the microbial stain is E. coli. And E. coli, there are a number of different E. coli stains that have been developed for different purposes in recombinant DNA technology applications. For instance, E. coli BL21D3 are routinely used for protein expression. This stain is deficient in proteases, lone proteases, and OMPT. Because proteases can degrade the protein of foreign protein, so genetically the genes, the proteases genes, are mutated or treated in these stains, and this which can increases the protein stability. While another strain of E. coli BL21 star, it is also a derivative of B3. It promotes the high messenger RNA stability, and which can lead to high protein yield. Similarly, BL21D3 PLYSS suppresses the basal expression, and it is ideal for toxic protein expression. Some proteins, fertile proteins, when expressed, they are, because they are not native to that bacteria or E. coli, that can be toxic. For those such proteins, this stain BL21D3PLYSS is ideal stain for toxic protein expression. And protein expression, soluble expression also depend upon the different vectors. And the choice of vectors actually depend upon desired protein. For example, if you're going to get higher protein expression, PET vectors are mostly used for high protein expression levels. And other than that, PET vectors have tight control over basal expression, and it is versatile and has many choices of tags and multiple cloning sites, and have convenient restriction sites for subcloning and one-step purification of heterologous proteins. Other vector which can be used is, there are actually a number of different vectors or plasmids like PAT28 and PSTV28, PTRC99A, and so on. And there is some vectors which works on can give good expression or low temperatures. That's called p-cold vectors. As their name indicates, these vectors improve protein expression at lower temperatures. And the protein synthesis rate. Protein synthesis rate, it's the multiple strategies can slow down the translation process for optimize protein solubility, because some sometime we need to slow down the process, but ultimately the slowing down this process for soluble protein expression can lead to low protein synthesis rate. So we can lower down protein synthesis rate by use of weak promoters, the use of low copy number plasmids, or lower growth temperatures, or by using the lower inducer of that vector, because good ideal vectors is only expressed we want when we induce it. Otherwise, it should not be induced. It should not be autonomous. It should be a controllable vector. So because the vectors are the burden on the cell, so unnecessarily we don't need to express those vectors. So by using the low amount of inducers, we can produce the low amount of the proteins. Higher the inducer, the higher the expression. The growth medium also plays important role in protein proper folding and protein stability. For example, addition of buffer in growth medium can control the pH fluctuation in the growth medium, especially when we grow the bacteria and big fermenters. We need to control the pH, and we use different buffers. Addition of glucose can repress the induction of the lac promoter by lactose, because glucose is easy to process and it's the universal uh, carbon source for bacteria and easy source. So we prefer glucose in a media. So we should not, because if there is no glucose, it can induce the expression of the lactose or prawn. So which can also hamper the protein expression, soluble protein expression. And then, for example, we can also add addition of polyols. It can work as a osmoprotectants and can stabilize protein structures. And other example is prosthetic groups or cofactors like trace metals, minerals, and vitamins, which can contribute to protein proper folding and stability. And then the fusion tags. Fusion tags is also called fusion partners, are used in E. coli to improve protein production yields, their stability and folding, and to facilitate protein purification. Fusion tag can also confer specific properties for target protein characterization and studies such as protein immunodetection, quantification, and structural and interactional studies. So the fusion tags are important for the different purposes. Either to give the stability to protein and to the characterize the protein. Some uh, a large variety of protein tags are available for improving protein stability and facilitating the soluble protein expression. Some of these tags, famous tags, are GST, glutathione or S transferase. GST, or which is also called glutathione S transferase, MBP, or the maltose binding protein, NASA, NUSA, or N utilization substance, or TRX, or thyrodoxine is tag, or polyhistidine tag, used for nickel column chromatography. So there are different tags for different purposes which can be utilized depending upon the design of your experiment and the protein express and your protein, which protein you are going to express. And the co-expression protein, in many cases, when proteins are expressed in insoluble aggregates or inclusion bodies, maybe they are not properly folded. Cell lacks the, for example, if there more protein is cell is overexpressing the protein and cell machinery cannot handle the excess amount of protein. For example, they can only, for example, bacteria can, for example, say at they can at the same time they can process the 100 amino acids and can fold in. But if you're expressing more and there are 200 or 500 amino acids came, and so it is above the threshold of those protein expression, protein folding, sorry, protein folding machinery. So it can results into insoluble invisible bodies, and because that proteins become insoluble. Usually soluble mean that the protein which are soluble in the aqueous phase of in the medium, but when they're not soluble they get settled as aggregates, so it's called inclusion bodies. And so protein expressed in soluble aggregate, it would be advantageous to co-express the protein with one or more other proteins. For example, if one protein is making aggregates and other protein is soluble, so we can combine those two proteins, and the soluble protein along with this protein which is initially insoluble, it can be soluble. Uh, the most important example in this case, the number one, the co-expression of proteins that form a complex. Often the separate components of a complex are not soluble, mostly due to the hydrophobic patches that are exposed to the solvent. For example, the different proteins in protein different amino acids. Some are hydrophobic, some are hydrophilic. That is, the amino acids which are facing towards the aqueous phase are hydrophilic, and like the 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 amino acids which are the hydrophobic, usually they are tend to surround by the hydrophilic amino acids. So there are different patches, which some patches in proteins are hydrophobic and some are hydrophilic. These patches are usually involved in protected by the binding to the other components of the complex. Therefore, co-expression of the different components could result in a higher expression level of a soluble protein complex. The co-expression of subunit of a di or multimeric proteins, or the co-expression of a one or more chaperones. For example, there are different in the different proteins. For example, if we have expressed protein, then proteins are only functional when they are properly folded. And for folding proteins, to fold our heterologous protein or the proteins which the cell will, they need proper folding. So for the proper folding, there's another mechanism of proteins and the system that's called chaperones or foldases. And these two classes of protein plays important role in vivo protein folding. So these are the factors which can be uh, influence the protein optimization in our experiments. But the main topic of my lecture is about the codon optimization. So we will continue on this codon optimization. So standard genetic codon. So what is standard genetic codon? Basically, we're talking about the protein, and proteins that consist of amino acids. And amino acids, the building block of the amino acids is different codons. So different codons make the amino acids, and amino acids make the protein, and the genetic code is genetic code specifies the amino acids. So the standard genetic code consists of the codons. The codon, what is codon? The three bases of DNA makes a codon. Actually, there are four DNA bases. So four DNA bases possibly can make 64 codons. These 64 codons code for 20 amino acids, including three stop codons. Why do codons matter? The fact. The first fact is why? The first fact is because according to the central dogma, genetic code provides a molecular expression for the transfer of information from DNA into messenger RNA and to the protein. And all of the genetic information depends on the forces and factors that determine how the nucleotide triplet code translates in the amino acid sequences. So basically, all of our genetic information is decoded in the form of proteins through genetic codons. So that's why genetic codes have their greater significance. So four DNA bases can make into 64 codons, and 64 codons can code 20 amino acids, different amino acids, because number of amino acids is 20. So as discussed earlier, that there are 64 codons, and each one codon is codes for triplet codon can code for one amino acid. For example, AUG here is can code for methionine, and then GUG is coding valine. The question is, there are 20 amino acids, but there are 64 codons. So why is there discrepancy? So it means that codon is genetic code is degenerate. So it means although each codon is specific for only one amino acid or one stop signal or one stop codon, genetic codon is described as a degenerate or redundant because a single amino acid may be coded for by more than one codons. So here's the explanation that there is 64 codons. But it means that more than that the amino acid may be coded by more than one codon. It is also important to note that the genetic code does not overlap. It means that each nucleotide is part of only one codon at a time. A single nucleotide cannot be part of two adjacent codons. And furthermore, the genetic code is nearly universal, with only rare variations reported. For instance, mitochondria have an alternative genetic code with slight variations. So codon degeneration indicates more than one codon can be used to encode an amino acid, and degeneracy in the genetic code enables a single protein to be encoded by the multitude of the synonymous gene sequences. And this degeneracy has a very important role in regulating protein expression. So there are different points where the protein expression can be regulated. For example, the DNA promoters, ribosomal binding sites, and then there are certain other factors which can influence the protein expression. But the main factors are promoters, ribosomal binding sites, and the degeneracy of these amino acids, and also has a great influence on protein expression. So in this chart, you can see that number of codons are coding the same amino acid. For example, in leucine is coded by the four different codons. In summary, an amino acid except methionine and tryptophan is encoded by 2 to 6 codons. So all the amino acids except methionine and tryptophan is coded by the 2 to 6 codons. So in continuation of this slide, the degeneracy of the genetic codon, we will discuss about the nucleotide substitution and protein coding genes. So actually there are two important substitutions in protein theory which can be divided into the synonymous substitutions or non-synonymous substitutions. For example, here in this diagram, you can in synonymous substitution, you can see that leucine is coded by the UAA, but in the below still it is giving leucine, but the codon is changed to UUG. A is replaced by the G. So, but the resulting amino acid is still the same leucine. Another example is threonine is coded both by the ACU and ACA. If ACU is replaced by the AC, UA replaced by the A, it still will produce the threonine. So the synonymous substitutions are point mutations which do not alter the amino acid sequences and are sometimes called silent mutations because they are producing that and they're producing the same amino acid. So if the amino acid does not change, then the protein is also unaffected. But the synonymous mutation can affect the protein expression rates up to the 1000-fold. So this is also another method of the protein fine-tuning the protein expression in bacteria. Synonymous mutations can also alter the protein conformation, post-translation modification, protein stability, and the function. Other mutation is called the non-synonymous substitution. In first example, that leucine is UUA giving the leucine, but if the A in the leucine is changed with the C, it will give into the phenylalanine and tryptophan UGG. If the G in the tryptophan is changed by the A, replaced by the A, it will result in the stop codon, which can result in the termination of the translation. Non-synonymous substitution non-synonymous substitution is a nucleotide mutation that alters the amino acid sequence of a protein. Non-synonymous mutation have a much greater effect on an individual protein than a synonymous mutation. It's also sometimes called nonsense mutation when it results in the stop uh, can result in the stop of the translation or the stop codon. In this example, the wild type DNA and this is the wild type messenger RNA. These are the amino acids. When mutated RNA, when there is stop codon, it there's no further increase in the protein amino acids. So here it is get terminated or it appears the stop codon. So actually this is the just for information, but related to our topic is synonymous substitution. You should keep in mind about the synonymous substitution. This means that if you change any nucleotide, but it still produce the same amino acid, it will not change your protein sequence. The protein will remain the same, but it can fine tune the protein expression and can affect on stability, conformation, and some other functions. So, codon usage bias. So, what is codon usage bias? Codon usage bias, what is this and how it is related to our topic we are going to discuss is one. Though a similar genetic code is used by the most organisms on Earth, but different organisms have different preferences for the codon they use to encode specific amino acid. For example, let's go to the previous slide. This means, for example, in E. coli, whenever leucine came, maybe it prefers the UAA for leucine, and for when threonine came, it prefers ACU, and for leucine, it mostly prefers UAA. But there are other also codons which can code for the leucine, but E. coli preferably, for example, prefers whenever there is leucine amino acid, it will use UAA for leucine. But for some other bacteria or some like yeast, when leucine came, maybe they prefer UUG instead of UAA. So different organisms have a different preference for the different codons. So this means they use different codons for the same amino acids. Uh, Different evolutionary constants have molded which codons are used preferentially. So as a result, this is the result of evolution on the longer period of time that different organisms have developed different preference for different codons. So it means evolutionary constants have molded which codons are used preferentially in which organisms. As a result, some transfer RNAs exist in very low quantities. Transfer RNAs mean that each amino acid codon is transported by the transfer RNA to the ribosomes. So if there is genetic codon is low in amounts, then their transfer RNAs also exist in very low quantities because the accessory codons are rarely used. Mean that is codon biasness mean that in a summary we can say that the some codons are more frequently used and some are used very less. Every species has codon usage biasness. For one species, some codons are rarely used, less than 1%, and these codons are called rare codons. So the codons which are used rarely or less than 1% in protein synthesis, those codons are called rare codons. So for example, rare codons in E. coli are different than rare codons in yeast or humans or Drosophila or plants or so on. Different organisms have different rare codons. So this is called codon usage biasness. Why do organisms have different codon usage biases? The reasons for varied codon preference organisms are not completely understood. But some possible results include is like metabolic pressures, the controlling gene expression through gene sequencing, protein folding, adaptation to the changing conditions. Briefly, I will discuss about the what is just for your knowledge metabolic pressure. What is metabolic pressure? How controlling gene expression, protein folding, adaptation to changing conditions. Metabolic pressure. What is that? Different cellular resources are required to produce transfer RNAs that requires different codons. Modify the transfer RNAs correctly and charge the transfer RNA with the appropriate amino acids. For example, if an organism uses only, for example, long time in using only a subset of codons, it only need to produce a subset of charged transfer RNAs. If they have some preference, they will frequently use that codons and it will not use others or maybe use very rarely. As a this results, their transfer RNA will be more and the amino acids which are used rarely, their amino acids will be in the low amounts ultimately. So if the organism uses only a subset of codon, it only need to produce a subset of charge transfer RNAs and therefore may need fewer resources for the entire translation process. For example, during high growth rate conditions, E. coli preferentially upregulate production of transfer RNAs that recognize codons found in highly expressed genes. Controlling gene expression through gene sequencing. Proteins that are encoded by the codons with low abundance or poorly charged transfer RNAs may be produced at a lower rate than proteins encoded by highly abundant charged transfer RNAs. For example, it was found that translation efficiency is well correlated with the codon bias in both E. coli and Saccharomyces cerevisiae. Then the protein folding. If the protein is encoded by a mixture of codons with a highly and poorly charged transfer RNAs, different regions of the protein may be translated at different rates. The ribosome will move quickly along regions calling for abundant charge transfer RNAs, but will stall or stops for a while at a region calling for a low abundant or poorly charged transfer RNAs. When the ribosome stalls, this may give the swiftly translated region a chance to fold properly. For example, in one study, it was found that tracts of non-optimal codons are associated with specific secondary structures in 10 closely related G strains. Then the adaptation to changing conditions. Adaptation to changing conditions, organisms often need to express genes at different levels under different conditions. It's not like that all genes are expressing at all the time. So bacteria are very economical while their expression. They only express those genes which they require. Other genes are not expressing. So they depend upon their need and amounts in which amount they need the expressions. With very codon fusion, an organism can change which proteins are highly expressed and which are poorly expressed by producing and charging specific transfer RNA pools. For example, transfer RNAs used in gene encoding amino acid biosynthesis enzymes may be preferentially charged during amino acid starvation, thus resulting in higher production of amino acid biosynthetic enzymes. Here are the examples of codon usage in E. coli. In this slide, using we can see that in this table we are AGG, AGA, AUA, CUA, CGA, CGG, CCC, UCG are the least used codons in E. coli. And codon usage was actually defined by Chang et al. In 1999 as the number of times, or if it is actually, he determined the frequency and defined the number of times a codon is translated per unit time in the cell of an organism. Actually, this definition is for real-time codon position, but it is hard to be measured in vivo. We use different algorithms and different protein sequencing, amino acid sequencing, gene sequencing in E. coli, all the gene, and then he determined out of those sequencing the frequency, and he found that these are the codons which are the least used codons of the E. coli. For example, AGG of arginine, AGA arginine, AUA isoleucine, CUA leucine, CGA arginine, CGG arginine, triple C for proline, and UCG serine. Here in this table you can see the 8 least codons used by E. coli is different from the yeast, Drosophila, and primates. So they have their own least used codons. So they are different. So their least codons of E. coli may be the most preferred or high codon in other organisms. So this is called codon usage bias. So they are biased in usage of the codon. Every organisms have different preference of the most using codons and the least using codons. For example, in arginine, E. coli, AGG, they use mostly they arginine, they use another codon. That codon present in the higher amount or abundance in E. coli, while these codons for arginine are are present rarely. The same is the case with the yeast. They have their own least and abundant codons. So and Drosophila has also different. In this table, you can analyze and can observe the least codons. They are different in different stains. So this is called the biasness. So they are actually amino acids are universal, but their preference is not universal. So their preference is there is preference and there is biasness among the organisms. This is the actually important point of this lecture. So why the protein we need to optimize the protein? What is rare codons? So this, if you got the idea from this slide, you will understand the whole lecture or the basic fundamental knowledge related to this tutorial. And this is another table, another example is that this is the usage of the 6 least used E. coli codons and in comparison with the other organisms. Here, if the codon frequencies of more than 15 per 1000 codons are shown in bold red. So, for example, in E. coli, arginine, AGG, the frequency, their preference is 1.4%, or on, on the other hand, Homo sapiens prefers this codon 11% or 11 times, and different organisms, for example, in Pyrococcus, the frequency is 30. So as compared to E. coli AGG is the least or rare codon, but or low abundant codon, but in Pyrococcus, it is frequency is 30, which is quite high. So arginine AGE, the for arginine codon AGA, it is 2.1, arginine CGA 3.1, leucine CUA 3.2, but for Plasmodium or Phyrococcus it is 18. So there are different preferences for the different organisms. For example, in case of proline, the frequency of triple proline in E. coli is 4.3%, but on the other hand, in Homo sapiens or humans, this preference or the frequency of usage of triple C for proline is 20. So this is the very good example of biasness. So E. coli don't prefer triple C for proline. It only prefers like 4.3%, while on humans they use like they prefer the 20% of or the 20% they will prefer this codon whenever there is proline came 20% frequency or 20 out of 100 20 times they will they will select this amino acid to incorporate in the prep for the proline. But in E. coli, they are rarely used. So this means E. coli, this proline is present in less abundant because they use it in low amounts. They will not prefer. So in the process of evolution, so bacteria they're using more amino acids they are produced in higher amounts that we produce only rarely they are present in low abundance and ultimately because the amino acids are coded by the transfer RNAs their respective transfer RNAs also present in the low abundance. So till here this is the first part of this lecture. Now we will move on to the second part this lecture in the next video.